All right, commissioners, thank you for letting me come here and speak to you today. I appreciate it. What I wanted to talk to you about today is give you an update on some of the wildlife disease research we have going on. A lot of it we've heard before, but hopefully it's new information and new progress on the research we're doing and how we're actually putting this research into action, improving surveillance, and improving management, and improving communication. So for those of you who are new or haven't heard this talk before, traditionally when we look at wildlife health and diseases, it's really been just a source of attrition in the traditional wildlife management. And it was sort of, disease was just one component that sort of kept wildlife populations in check. And oftentimes they felt, well, things will just run its course. But over the last 1 to 2 decades, we've seen multiple diseases emerge that have significant implications for wildlife health, for human health, for domestic animal health, and have required our agency or other wildlife agencies to actually conduct surveillance and manage some of these diseases. So for wildlife health, in regards to how we monitor diseases in the state, we take an integrated approach because we don't always have, depending on the species we're dealing with, the best tools to identify disease, to monitor disease, and more specifically to monitor their impacts. So again, we take sort of an integrated approach and try and focus and look at disease from a variety of different angles. Certainly we can look at the literature. If there is any research or surveillance that's been done in other states, we try and incorporate that information so that we don't have to repeat it, and that's a good starting point. We look at population data to see how those populations are doing relative to the occurrence of disease to give us an idea of whether the disease is actually having impacts on the population level. We can conduct surveillance for disease, and we can do this sort of 2 broad ways. One we can call passive surveillance, which is simply our disease investigations. So throughout the year, we get reports from the general public, hunters, that are calling in sick or dead animals. They collect, our field staff can collect those animals, bring them to me or another lab, and we can identify disease that way. But certainly we know that disease may be hard to detect in some wildlife species. If they're sick, they may go into the woods where they're never found. They can be scavenged or predated upon, and so we can't get those samples. And in some cases, wildlife can harbor disease without clinical signs at all. So we also have this second category of surveillance, which we call active surveillance. So that's where we actually go out, collect biological samples from wildlife, regardless of whether they're sick or not, and specifically look for those diseases. So that's what we do with CWD when we're pulling those heads out of the barrels, collecting samples and testing for those prions. And then the final component of our wildlife health program is applied research, where we try and take all these other categories as well as doing research to identify what's going on, is there any way we can manage or mitigate these diseases, and what are the real impacts. So I'll give you 2 examples here on sort of how we're pulling all those different facets together for 2 projects. We'll look at West Nile and mange, and then briefly mention some of the other diseases that we have coming down the pike. So when we look at West Nile and ruffed grouse, and we've talked about this previously, when Lisa and I first started to look at this problem in 2014, on the surface, it didn't look like it was going to be that challenging. We're dealing with ruffed grouse, obviously the state bird, a really popular game bird species, one that we have a very passionate hunting community that try and hunt grouse. So we have a very invested hunting community in this species, and it seems like we would have a lot of data on them. And certainly we do on their population. But if we look at our data on disease and health, really, there's very little on grouse in Pennsylvania or nationally. There's just not a lot of health and disease data there. So we had sort of a knowledge gap. When we look at West Nile, it's one of the most well-studied viral diseases in the last 20 years. It emerged in 1999 in New York City suburbs, and over the next few years spread throughout the country, and it's now throughout the lower 48. It's been a variety of research done on impacts on human health, domestic animals, if we look at horses, and even in wildlife species, particularly on the corvids and raptors. But most of the research that's been done in West Nile has been done by public health agencies. And for that reason, it's really been focused on those urban, suburban centers. And so if you took sort of a graph or a map on where West Nile research has been done to date, It's almost the polar opposite of where you're going to find grouse. So again, we had another knowledge gap there of we have a lot of information on this, but we don't know what happens with West Nile in the woods. So we started out with these flush rates. Again, this would sort of be our population data. And we can see in here, and Lisa's shown you this graph before, this sort of classic boom or bust cycle where that dotted sort of horizontal line is the long year 50-year average. These are our fall flush rates. We basically collect these by a large hunter cooperator group that keep diaries, log their hours of where they hunt, whether they bag a bird, and a variety of other information. And using that data, we can piece together this sort of abundance data based on these fall flush rates. And again, you can see sort of this boom or bust cycle where the grouse populations go up and down until around 2000, 2001, when you see that dramatic decline. It does rebound, but never really goes above that long year 50-year average. And again, within that area that's pink, you can see we've got about 5 or 6 of our record lows for grouse populations in Pennsylvania. And this pink area here basically represents when West Nile arrived in Pennsylvania and spread throughout the state. If we look at it a little bit closer, sorry that didn't show up. If you look at it a little bit closer, we can look here on this figure on the left. The red is again going to be those fall flush rates. And the orange and yellow represent West Nile surveillance that's done here in Pennsylvania. So in Pennsylvania, West Nile surveillance is conducted by the Department of Environmental Protection. They do this both by monitoring for dead birds. So if someone finds a dead corvid or raptor, they can submit it to DEP to be tested. But they also use these mosquito pools where they go out and throughout the state, they trap mosquitoes in urban, suburban centers. They test them, they pool them, and that gives a more systematic way and more sensitive way to detect when West Nile is happening in the state. And you can see, looking at this figure, that when it's a bad West Nile year, whether in dead birds or in mosquito pools, you can see that population decline. In years where West Nile isn't so bad, the population can go back up until you have the next bad year, and the population can decline again. So again, if we use that population data that we have, almost all of our metrics in grouse show that we have negative associations with the occurrence of West Nile. But certainly that's not enough to fall back on completely. So we went to the next step and we conducted a challenge study. To do this, we collected eggs from the field. We hatched out the eggs in captivity, raised them in confinement so they know they wouldn't have been exposed to West Nile. We then moved them to Colorado State where we challenged them with West Nile virus. And what we found is out of the 10 birds that we experimentally infected, 40% became severely ill, had to be euthanized, or died within 7 to 8 days of exposure. The remaining 6 had moderate to severe lesions in the heart or in the brain. So just about every bird that was infected was severely or at least moderately affected or died from the viral infection. And when you compare those to what's been done in other species, it suggests that they're highly susceptible to West Nile. So again, we had the population data, we had the susceptibility data. And this is just a paper that we published off of this last year in veterinary pathology. So we know that we see the population impacts, we see the susceptibility. The next question was, are grouse even being exposed? Because potentially we could be going down sort of the wrong route with this if they're not even being exposed in the woods. To do this, and when we look at exposure, we basically collect blood samples. It's not really feasible to get a usable blood sample from a hunter-harvested grouse just because you need to collect it pretty quickly and process it. So what we used instead were these sort of filter paper strips, these sort of T-shaped filters here. And basically what you can do with these is just soak it in the blood, bring it back to the lab, and pull antibodies off of it. And here you can see one right here that's sort of blood-soaked. And this gave us an easy way to reach out to our hunter cooperators and say, look, we know you're part of our cooperator. We actually were able to target grouse hunters in certain areas, and we sent out about 600 of these. And we said, if you harvest a grouse, collect this, soak it in blood so that you can get those antibodies on that strip, collect feathers that we need for aging and sexing, and then after you're done with the season, let it air dry, or after you're done with the hunt, let it air dry and then send it back to us. What we were able to do is over 2 years collect over 400 samples, 418 samples, and you can see the distribution here in this map. So a really good return rate from this effort. We were able to show that grouse are exposed to West Nile in all 6 parts of the state. We see our highest prevalence of antibodies up here in sort of the classic grouse area in that north-central northwest. We did see variation in seroprevalence. In 2015-16, it was about 14% of our birds had antibodies. In 16-17, it was a little higher at 24%. We did continue this last year, which was one of a really bad West Nile year, and those samples are actually still being tested at Colorado State. There's been quite a lot of interest in this antibody survey, and we're currently analyzing our data with Squidus to look at what patterns may be able to account for why we're seeing exposure higher in some areas versus others. And so that analysis is still underway. But we've received interest from many states throughout the grouse range, ranging from eastern Canada down to Georgia and in the upper Midwest. So there's actually currently a movement to have sort of a range-wide antibody survey being conducted throughout the grouse range, and we hope to do that this winter. So again, we've got exposure, susceptibility, population data. The next big question we had was, well, what's happening in the woods? When we reached out to the grouse the West Nile experts, and we said, what do mosquitoes look like in the woods? What does West Nile transmission look like in the woods? There were 0 answers. So we started out last year out at Scotia, and Lisa and I worked with DEP, who have been amazing collaborators on this, and we selected 8 trap sites for mosquitoes throughout Scotia, representing different grouse habitat throughout their life. We went there with DEP, and Lisa would select an area, and then the DEP would come in and say, okay, if you want to be here, this is where you put the trap, and this is where you can get the mosquitoes. We trapped mosquitoes starting in June and ending in September. DEP was actually, for free, identified all the mosquitoes that we trapped and actually did testing for different pools of specific species for West Nile. So again, amazing collaborators on this. Over that period, we trapped over 15,000 mosquitoes, 25 different species, and 8 that fit into the category of they were competent vectors of West Nile, so they could carry that virus, as well as they fed on birds. And one of those that really popped out was about 22% of our total samples was Culex restuans, and that's this mosquito right here. And this is pretty interesting because when you look at West Nile in urban-suburban settings, you see different species of Culex that are actually transmitting West Nile predominantly within those habitats. So it does look like a different driving or predominant mosquito is involved in this wooded habitat. We did have 3 pools that tested positive of those mosquitoes for West. Interestingly, again, all of them were that same species of mosquito, Culex restuans, and the positives that showed up in our trapping in Scotia aligned really closely with when they were getting positives in urban and suburban settings by DEP. So what we're doing now, again, we've completed the population analysis, looking at that population data and how it relates to West Nile. We've done our experimental infection study, the statewide antibody survey that we're currently analyzing and going to publish something on later this summer, hopefully. And the current ongoing studies are, one, that regional antibody survey. So again, multiple states are interested, and we're going to do basically like an exposure of West Nile and grouse throughout the range of grouse, looking at different patterns and different things that may associate with infection and exposure. We're also expanding our mosquito surveillance. We've selected 7 sites throughout Pennsylvania, and we're not only looking at, again, that same question of what mosquitoes are biting in the woods, but we're also looking at different factors that may influence our management, looking at elevation gradients to see if that can influence mosquito activity and West Nile activity. We're looking at distance to roads and things like that, because we know things like tire ruts and other things like that are good environments for mosquitoes. So we're looking at a variety of different factors related to this mosquito surveillance. So what have we done with this data? Certainly we've improved our surveillance. We started out saying, I don't know how we figure out how we're going to get West Nile or detect West Nile and grouse. But we now know how to detect it better in individuals. What do the lesions look like? What are the best samples to collect to identify the virus? We know how to identify it better in the population through this antibody survey using those filter strips. And we're currently looking on better ways that we can identify it in the environment, and even seeing if we have to, because ideally what happens in the woods mirrors what happens in the urban suburban centers by DEP. And if that's the case, then we can just use their data moving forward and saying, yes, this is a bad West Nile year, or no, we have to collect our own data. We've obviously used it for management, and Lisa's presented this before, creating the adaptive harvest framework, as well as sort of having this being even more emphasis of why we need habitat, and also in some ways looking at ways that we can create habitat in a more efficient way. If we do see elevation gradients, if we do see distance to roads or other factors that are involved with mosquitoes. And we've also used it for communication, not just in Pennsylvania, but they're using this research nationally. So last year was supposed to be a record year in the upper Midwest for grouse, and unfortunately it was one of the worst years they had. And without an explanation, they were sort of left wondering what might be going on. It turns out it was also one of the worst West Nile years they've had. And when they went back to their diagnostic data, they had record numbers of grouse submitted for diagnostic cases. So a lot of the work we're doing here provided explanations rather than, we don't know why it was a bad year, it should have been good. We're also using this, and I don't know if we're ever going to get to the level of being predictive. Unfortunately, West Nile is just too unpredictable to be able to predict what's going to happen in a given year. However, we do know if we have a bad West Nile year, what that, at least historically, has done. And unfortunately, this year, we're on pace to being one of the worst West Nile years that we've had in Pennsylvania history. So again, hopefully that trend does not continue, but unfortunately, it's looking like a really bad year. So the second project we've been really active on recently is mange in bears. And again, what mange is, it's a generic term for a contagious parasitic disease of the skin of animals. In humans, it's called scabies. It's got a characteristic set of clinical signs regardless of the animal species. Typically, it results in itching and scratching and extreme discomfort just from the itchiness of the skin. It can lead to behavioral changes where the animal's acting erratic just because they're so itchy. You eventually lead to hair loss and variable degrees of crusting. There's a foul odor to it, often with secondary infections with bacteria and yeast. And then eventually, as the disease continues and you get hair loss and other issues, you get a loss of body condition. So oftentimes, you have animals that look like this here, where they're emaciated, have crusty, scaly, scabby skin, and obviously are in moribund condition. Historically, when we look at mange in bears, it's been a real sporadic disease. It's been one where you may see an individual animal have it here or there, but typically it's just an individual animal thing. Unfortunately, the situation is a little bit different here in Pennsylvania, and we first started to see increasing number of mange cases in sort of the central western part of the state in the late 80s, early 90s. And then over time, we can see it's expanded outward from there and has currently gone into adjoining states, into New York, just over the border, as well as Maryland, West Virginia, and Virginia. Now, this map oftentimes is misinterpreted, and the way you read it is basically the first time a township goes positive, it turns red, and then it reverts to gray. But what is often interpreted is that we are just seeing mange raging throughout this area here. What actually happens, and what we're going to start to look at a little bit more, is sort of the cycling that happens in the endemic area. Because within this area here, you've got sort of hot spots that come and go within those areas. So I think a lot of times people see this map and they just think there's mange everywhere in Pennsylvania. But what actually happens is you'll get an area like right now what we're seeing in sort of Center County and some other parts of North Central where it's really bad for 2 to 3 years, and then it may sort of die down and go away for a while. But it sort of cycles within this area. But certainly, it's not just an expansion of range. As you get that range expansion, we also see increasing number of cases. So again, this sort of leads to our passive surveillance, where are we seeing sick and dead animals? And we can see whatever way we measure it, whether it's euthanized animals because they're moribund or sick, animals with mange showing up in the harvest, animals that are trapping as part of our research trapping that have mange, or roadkills. In all of those different measures, we're seeing increasing number of mange cases. And so certainly when we look at mange, this is what we see, and we think, okay, mange is an increasing problem in the state. And I don't think that's an unfair assessment to make. But part of the thing we're seeing, much like we would imagine with lead, is that you're seeing increasing cases, you're seeing an expansion of disease, but we're also seeing an expanding bear population. So while I think it's fair to say that mange is an increasing problem in the state, when we try and quantify that and say, well, what about prevalence or something like that, it's that we don't exactly know at this point, because we know it's increasing, but so is the bear population. So it's one thing that we're currently looking at a little bit more to say, how can we properly quantify how much of the increasing mange is just an increased percentage of disease versus an expanding and growing population. So we started this about 3 years ago with a variety of questions trying to better understand mange in the state and why Pennsylvania is unique. We started out with an easy question of what's the mite species that's causing mange, looking at ways we can identify it better in bears and confirm infection, looking at the environmental survival, and then finally looking at the survival of bears with mange to see how many of them that have it actually succumb to the infestation. So looking first at the causative mite, if you look historically, there have been 3 different mites that cause mange in bears. There's Sarcoptes, Ursicoptes, and Demodex. And it's important to characterize these different ones because each of those mites have different characteristics. Ursicoptes, it's only in bears, only causes disease in bears. Sarcoptes can cause disease in a variety of species, including humans. Demodex, it's only bear-specific. So it was really important that we identify what's the mite we're dealing with. This is 2 papers that came out of this work, working with the University of Georgia. Basically, the way we addressed this was we had 99 bears from 2015 to 17 that had severe mange. We scraped them and identified what the mite was in those. Out of the 99, we had 91 where we identified mites, and in 100% of those, it was the mite Sarcoptes scabiei, which is this mite shown here. There's its mouth parts, its front legs, and sort of these hairs coming off its legs there. Sarcoptes scabiei is a mite that has a very wide host range. It's been identified in a whole host of different mammals, depending on where you are. Here in North America, we predominantly see it in our canids. So if you see a mangy coyote, a mangy red fox, a farm dog, that's Sarcoptes scabiei. If you see humans with scabies, that's Sarcoptes scabiei as well. The thought is that they have, although they're all Sarcoptes scabiei, they have different host strain variants. So one strain is adapted to bears, one's adapted to dogs, one's adapted to humans. It can infect each other, but the bear mite won't do as well on dogs as it will on bears, so that there are some host adaptation there. The mite lives within the upper layer of the skin, so this is a little cartoon of what your skin looks like. It creates these tunnels. The complete life cycle in the skin, so it lays eggs, they go to the surface, and reproduction and everything happens on the same host. The mite burden can be really high, so you can imagine how they're all reproducing here. You can get up to thousands of mites on those skins, on that skin, and that just facilitates transmission between animals. So the next question was, how do we diagnose it in bears? And this was important because we get a lot of pictures of bears with mange, right, or bears with hair loss. Some of those are mange, some of those are other things. It can be anything from an atypical hair coat change. It can be hormonal diseases. We've seen a whole host of different things that cause hair loss in bears. So if we're going to start monitoring it, we have to be able to confirm it in individual animals as well as the population. We looked at a variety of different ways of how we can identify mites and what's the most sensitive tool that we have to do that. We looked at their genetic material, scraping, doing biopsies, looking at any sort of mite DNA there. And it turned out that the most sensitive way to actually detect the mite was simply scraping the skin and looking at it under a microscope. And so now for some of these subsequent studies, we've brought little small microscopes into the field, and if we have a bear that's down, we can scrape its skin and have a diagnosis right there, rather than having to bring samples back to a lab and do other sort of things that may take several days. So we've got the abilities now to do a rather quick, easy, and relatively inexpensive way to confirm it in individual bears. We also wanted to identify it in the population to get at that question of, is the prevalence of mange increasing and to what extent is that changing over time. For this to be reliable, again, we'd have to go to an antibody test. What antibodies tell us is that the animal's been exposed and survived. We looked at a commercial antibody test that's available. It's actually based on Sarcoptes scabiei in dogs. But when we validated it and evaluated it in bears, it turned out that it worked really well. So of the 45 bears that we had confirmed infection, they identified antibodies in 92% of them. And so for an antibody test, that's actually pretty good. We also looked at cubs from sows that didn't have antibodies, and none of those were positive. So in our initial stages, it looks like our antibody test works really well. And so it's something that we now want to sort of go out into the field and look at mange exposure in our population of bears. The next question we looked at was environmental survival. So if we have a bear with mange, is it always have to be direct contact, or can you get mange from them using the same location? Can it be transmitted through the environment, through dens, through trap sites, things like that? So for this, we collected large patches of skin from bears immediately after they were euthanized, sent those down to University of Georgia, and a grad student there basically put them at different temperature chambers, at freezing temperature, winter temperature, which would be refrigeration at 4C, more spring-like temperatures, and then summer temperatures. And he basically monitored the survival of those mites every hour, and then eventually every day until they were no longer alive. And what he found is that, and here you can sort of see looking under the microscope, there's a little mite right there, there's a mite right there. So this student basically just sat there and looked at mange skin for hours at a time for several days. And thank God he did, because it actually gave us usable data. So what we found, and we're still running a few of these trials to get this data together, but he found that freezing actually inactivates the mites. So if we get a skin or a carcass or anything like that, one freeze-thaw cycle will inactivate those mites. When we get above freezing, the survival of the mite depends on the temperature. So at 4C, which is our coolest temperature, they can last anywhere from about 6 to 12 days. If we get to more like summer temperatures, we're looking at a survival of around 1 to 4. So what we've started to do is generate these survival curves where they basically show us at different temperatures how long these mites are going to be able to survive. And this is important. It tells us a few things. One, we're not dealing with an environmental reservoir. So it's not like CWD where if a mange bear comes into a yard or an area that those mites are going to be there forever. They're actually not going to live that long. In the summer, we're probably looking at a few days. In sort of those cooler spring-like temperatures, we're probably looking anywhere from about 1 to 2 weeks. It also tells us that freezing will inactivate them. So if we have someone that has a carcass and is concerned, or if we have freezing temperatures, that's going to inactivate those mites that are in the environment. So when we go out, obviously there are concerns. When we have things such as feeding or anything that congregates bears, oftentimes you'll hear people say, well, there aren't bears coming at the same time, so I'm okay. But what this tells us is if you've got a mangy bear come into a feeder or come into a garbage can, whatever comes to that garbage can or feeder over the next 7 to 10 days, depending on the temperature, could potentially be exposed to these mites. So for the mange studies, what we've done, we've completed sort of the identification and genetic characterization of the mite that's causing mange in Pennsylvania. We've identified ways we can test both populations as well as individual bears to confirm infection. We're finishing up an environmental study and are currently writing that up for publication now. We're going to now start an antibody survey, so to give us sort of an idea of what is the prevalence of mange in different parts of the state. And that'll be a measure that we can use to follow over time to say, is it getting worse? Is it cycling to different parts of our endemic area like we see with our mange cases or not? We've currently started a new study this summer with Penn State University, where we're actually radio collaring bears that have mange that have a certain parameter of mange. So if it's severe mange, those animals are still humanely euthanized. But if it's not severe, we are putting and can confirm it microscopically in the field. We're putting collars on them and following them over to look at survival, to look at activity, over time, both for treated and untreated bears. And hopefully that'll give us a lot better information on the impacts of mange on individual animals as well as our treatment efficacy. So what we've done with this data, we've certainly improved our surveillance. We now know how to confirm it in individual bears, so we're not dealing with situations that every bear that has a bad hair day we're calling mange. We've looked at how to identify it in the population, both identifying tools as well as a way that we can monitor hunter-harvested bears and sort of have a systematic way we can monitor it in populations over time. We've used the data, particularly the environmental persistence data, to refine our SOPs, or at least that's what we're going to be doing this fall, to give people better ideas of if you have a mange bear show up in a trap or a trap site, how long do you have to leave that dormant under different conditions, and what can you do with a carcass to make sure that we're not contributing to mange in Pennsylvania. And certainly communication. This year we had an SDIC training in the North Central region on feeding that we're currently doing. And this is the kind of information that I think they can really relate to, where we've got an increasing problem and we've got actual concrete data to show how long these mites can live in the environment and how that can relate and impact feeding and the health of bears and other wildlife. In regards to other active projects we've been working quite a bit with wild turkey diseases, with active projects on lymphoproliferative disease virus, or LPDV, some work with blackhead as well as pox. We've worked with lead toxicity in bald eagles, which I presented last fall, and we've moved on with that even further to work with Cornell to look at sort of some of the metadata over the entire East, looking at lead impacts on bald eagle health. Certainly chronic wasting disease, white nose, which Greg will present to you all next month, some of the really great work he's been doing. This summer we initiated our muskrat health and disease project, Matt and I, as well in collaboration with Penn State, looking at muskrat survival with intraperitoneal transmitters, and also doing a little bit of work on health and disease in muskrats. We've completed a 4-year AI avian influenza study characterizing the epidemiology and ecology of viruses that ducks migrate through Pennsylvania with. And more recently, we've initiated surveillance for the longhorn tick here in Pennsylvania, which is a new invasive tick that first showed up last year in New Jersey and has now been detected in 9 states in the East. So I'm happy to take any questions you have on this or any other wildlife health projects.
So when we're looking at the grouse populations fluctuate normally. So did you see, because I was trying to figure it out on your chart, when the grouse populations were low, is that when we got hit with the first West Nile, and you think that's why the populations are staying below the average?
So,
you follow me?
You know, right here, I think we could come up with a whole host of things that may have influenced it, and Lisa would probably be able to speak to that. But this is certainly even before West Nile was in North America.
But I'm talking even like in 99.
This would be in the mid-70s here. Starting right here is 2000. So this is when 2000, 2001, when West Nile hit throughout the state. Then you see that decline. You do see a recovery, but it never really gets above sort of that long-term 50-year average. And then in the last 5 to 10 years, it's been well below. So it's got that boom or bust cycle still, but it's now shifted down. And part of the problem is this is good, generally skewed towards good habitat, because these are fall flush rates, so hunters aren't going to go out to places where grouse aren't doing well. So this is going to be skewed more towards good habitat. If we break it into sort of north, central, northwest, we do see the declines, but they do rebound. So if we go to places where they have good habitat, those populations will rebound if we have time between West Nile outbreaks. If we go to areas where we don't have that habitat, so most of the south of I-80, what we see is it'll decline, a very weak recovery until it declines again, and it'll just be sort of a stepwise pattern down. So we sort of have to look at it under two different habitat types.
Right.
What about all the other bird species?
That's a great question. What about the other bird species? There's a great paper that came out and was published actually in the Proceedings of the National Academy of Sciences that just sort of listed a bunch of migratory passerines that they felt could have been impacted by West Nile. But we just don't have that data to confirm it or refute it. Unfortunately, a lot of what we know about with West Nile in wild birds, again, is going to be skewed towards those urban-suburban settings. So it's going to be your corvids. It's going to be your raptors that you might be able to find. So if something small dies in the woods, we just are rarely ever going to know about it until it's potentially a really big problem. So there is increasing movement towards looking at West Nile in other upland game birds. So looking at it in specifically turkeys and quail are the large focus now, but even potentially woodcock, just because we now have at least 2 galliform species that are highly susceptible, being sage grouse and ruffed grouse.
Any other questions? Great, thanks, Justin.
All right,
Appreciate
thanks.
it. Yeah, I'll know who's up, so you don't even have to call.
Okay, good afternoon. My name is John Wakefield. I'm the acting division chief for Habitat Planning and Development. I'm not John Toucher, but John Toucher is the private land section chief. He couldn't make it today, so I'm here filling in. And what's being passed around is some outreach materials that we'll talk about here in a little bit. We're trying to ramp up our private lands program, and that's what this presentation is about. Okay, we'll start off the presentation with our agency mission statement. Manage Pennsylvania's wild birds, wild mammals, and their habitats for current and future generations. And it's important to note that habitat management is recognized in our agency mission statement. Seasons and bag limits is a tool that we can use to manage wildlife populations, but it's not enough by itself. We need to have quality habitat, appropriate habitat to meet our goals for wildlife management. Pennsylvania is home to 480 species of wild birds and mammals, so you can just imagine the diversity and differences in habitat that we're going to need out there across the landscape. Of course, we manage 1.5 million acres of state game lands for wildlife. DCNR is managing state forests and state parks. There are several federal lands out there, National Forest and National Park, and they have some wildlife objectives. And this is all public lands management, public lands access to hunting. But here's the problem. That's only 15% of Pennsylvania's land base is public lands. The remaining 85% is privately owned. So we need to focus on private lands management in order to meet our mission. And private lands management does come with its challenges. You know, all the parcelization that's out there, a bunch of landowners that we're going to have to work with. And this issue is not new. We've recognized this in the past. We have several goals in our agency strategic plan that are particular to private lands management. One of those being the need to increase grassland and young forest acres. We wanted to get an additional 5,000 acres on private lands within the strategic plan time period, and we've already surpassed that within its first couple years. So the state and federal agencies are working on habitat management on private lands. As I mentioned before, the private lands piece has several challenges, one of those being different landowner goals and all that, the small parcelization, trying to work with all those multiple landowners out there and the different land uses, and ultimately trying to get landowners together to come together on a common mission. And whether that's across boundaries of private lands and public lands or just multiple private landowners, if we really want to have a meaningful impact in the state, it's going to be trying to get landowners pulled together as we go forward with some of these activities. State governments and NGOs are already working on some private lands initiatives out there. So again, this is not anything earth-shattering new. We'll get to our for taking things in a new direction here going forward. Aside from some of the parcelization concerns and the overall structure of the private lands in Pennsylvania, we face some ecological challenges. So forest age composition, forest health, farmland, and water quality. And I'll get into discussing each of those here in the next couple slides. So forest age. About 75% of Pennsylvania's forests can be considered mature. So we're looking at that old age class. Everything was cut over the turn of the 19th century, all grew up all at the same time, and we've had pretty little impact statewide since then in creating young forests. I mean, as a whole, we certainly had initiatives out there and are working at it, but this is the current state of the forest. 75% is classified as mature, and for quality wildlife habitat, we need young forest. We need that structural diversity. So creating that poses a challenge. Several partner opportunities that we have out there. National Wild Turkey Federation, the Ruffed Grouse Society, Wildlife Management Institute, and American Bird Conservancy all have their own initiatives to create young forest habitat, and they also come with their own funding sources, which we can tap into and work partnerships. Forest health. So we have various insects and diseases that are a challenge, invasive plants, poor forest management, particularly an issue on private lands. It's been an initiative of DCNR, Penn State Extension, to try to help landowners do good forest management. But as you can see on the next bullet, that costs money. And as a reality, there's not a whole lot of landowners who are going to be willing to shell out additional money to maybe do what's right to combat some of these insects and diseases and invasive plants and do proper forest management. So partner opportunities out there for this: National Wild Turkey Federation, DCNR, and NRCS. Again, all have initiatives focused on quality forest management, and they have additional funding opportunities there that we can work as partners. Okay, farmland. So two main points here. Development. So loss of farmland due to residential housing development and industrial commercial development. Particularly here in Pennsylvania, warehouse development along the 81 corridor, I-78 corridor is pretty huge. We're seeing all kind of farmland be developed and go under concrete for warehouse development. In the farmland that we do have, clean farming, intensive farming is a problem. There's no longer weeds in our cornfields, and once crops are harvested, there's not a whole lot left there to benefit wildlife. So partners that are working to improve habitat on farmland include Pheasants Forever, Chesapeake Bay Foundation and NRCS, and a notable program here is CREP, that NRCS has. And then we have various land trusts out there that are trying to preserve farmland habitat from development. And then water quality is a major issue. 84,000 miles of streams in Pennsylvania, 16,000 of those are listed as impaired. We have 400,000 acres of wetlands, and that's at a 56% loss of what was historically here. So some major challenges here due to development, agriculture impacts, invasive species, and erosion. And so partners that we're working with on some of these initiatives, Ducks Unlimited, Trout Unlimited, Chesapeake Bay Foundation, and NRCS. So given all that background, we'll talk about our approach and our situation with private lands habitat management. We are one of few state agencies across the country that does not employ private lands biologists. With our decreased staffing levels, we're having to rely on private lands as another duty as assigned to other staff. So that's biologists, game wardens, land managers, habitat crew, amongst everything else that we have going on. We throw this private lands piece in there and have to make do with that. That's just the reality. A big ticket for us is our hunter access program. And that's where we're going to put our focus here going forward with trying to do more on private lands, is our ability to work on our public access cooperators. As a game commission, it gives us the ability to work on those properties, and it also provides a designated funding source. So we have VEPA HIP funds that can be spent on these hunter access cooperators. So really to think back to that slide, that large land mass of Pennsylvania, 85% of our land is privately owned. It's such a big task. It's pretty darn overwhelming to try to think about, okay, how are we gonna get started here? How are we gonna make a meaningful impact? Let's narrow it down. We can work on hunter access properties, get things started, and really get this program up and running. All right, so here's a look at the access program. We have 13,000 landowners enrolled, and it gives us roughly 2.1, 2.2 million acres of habitat. And we've seen an increase of about 161,000 acres since the start of the strategic plan in 2015. And that was another goal in that the plan was to increase the cooperation in our hunter access program. As you can see the disbursement across Pennsylvania of all those enrollments. We have a large variety of habitat that's enrolled on these properties, some pretty unique opportunities that we don't have on game lands in some circumstances. A dedicated VEPA HIP funding, so it's volunteer public access habitat incentive program, comes out of the Farm Bill. It's funding that's dedicated for use on hunter access properties. So this program, just a little bit about it. Landowners sign up for a 5-year commitment, and then if we do habitat work on that property, it turns into a 10-year commitment. So we get some longevity there for the work that we're doing. We have dedicated funding again, so the landowner gets free habitat work done. They get subscription to the game news. They get free tree seedlings, and they can get some increased law enforcement presence for any issues that they might have. So lots of positives all the way around for both us and those private landowners. And we'll scale up or scale back those projects based on funding, depending on how much we have at a given time. So some specific goals for the program. We found in a recent survey that a large majority of landowners had no clue about this hunter access program. The knowledge just wasn't out there. So one of our big initiatives is to get messaging out to the public, get some workshops put together, do some field days, do some presentations, just to educate. We intend to do between 500 and 1,000 acres of habitat improvement annually over the next 4 years through VEPA HIP money. And we want to line up a queue of at least 1,500 acres of plans, have them ready to go in case additional funding comes available. We don't have to jump through the hoops and try to line up projects at that time. We'll have a queue sitting and ready to go so we can jump on that opportunity. And we're going to look to combine efforts with other organizations, continue and build partnerships, which will allow us to have an impact on lands that are not enrolled in hunter access as well. So there's partnerships with Pheasants Forever, NRCS, NWTF, things like that. Okay, I want to highlight one partnership that that'll be a new partnership for us. We're going to enter into a contract with Habitat Forever, which is an affiliate organization of Pheasants Forever. And we're going to fund 4 positions that will focus on habitat planning, coordinating, implementation on hunter access properties. We're going to staff positions in the North Central, Northeast, South, and Southwest regions, and then 1 position out of Harrisburg. And those people will be available statewide for this habitat work, but particularly in those named regions. And that's going to provide us some stability in implementation. Those folks are going to be able to actually do some small projects on the ground, but they'll also have that primary focus on implementing private lands work, and will provide a pretty steady funding stream for implementation and contracting. We're going to look to continue existing partnerships that we currently have going on. Farm Bill biologists in a cooperative agreement with NRCS and Pheasants Forever. We have 5 of those across the state right now. We may be able to bump that up by 2 or 3 additional here over the next year or 2. And they're implementing CREP on private lands. Working Lands for Wildlife, that's an initiative focused on golden wing warbler habitat. And then RCPP, Regional Conservation Partnership Program, focuses on cerulean warbler habitat. So species-specific initiatives, they've been ongoing. We've been involved with these for several years now. They come with some funding of their own, so we'll look to continue that and ramping those projects up. Okay, and of course we plan to continue expanding the private lands program in the future. A big part of this is going to be coordination with the Bureau of I&E on development of brochures, materials, a communication strategy, some of these workshops, to try to get that messaging out there. And we're going to look to secure additional funding for the private lands program. 2018 VEPA HIP grant, again, that's tied to the Farm Bill. The Farm Bill may pass this fall. They're still trying to work through that. But we'll be jumping on that as soon as things get finalized. And then we'll look to increase our partnerships, again, with those species-specific initiatives, golden wing warblers, cerulean warbler habitat projects, and then habitat-specific programs, young forest initiatives, things like that through American Bird Conservancy and other NGOs out there. Okay. Take any questions?
Are there any questions from the commissioners? I don't think. Thank you much.
Good.
Do we need a break? Danny's leaving. I don't know. I think we just got started. Take a break? Let's take a 10-minute break since our new commissioner is leaving us. 10 minutes, promptly back here in 10.